about genetic code. Have you read genetic code? If it is not, if you have not read, it will be very difficult. There is a full class on genetic code. I mean, those who have read, for them I will show a few slides that will be very important, particularly one or two areas where MCQ questions might be set. You just note down the idea only, okay, and the salient points. So genetic code, why deliberately I am telling, because genetic code is very required for translation. Without genetic code, translation cannot be done. So genetic code, basically definition, all these things I am not telling because this is not a class on genetic code. Basically it is a 3-letter combination, nucleotide combination, triplet combination, that is basically genetic code, and there are 64 codons, 61 actually codes, and 3 codons are for soft codons that you must remember: UGA, UAG and UGA. UAA, UAG and UGA. That you must remember. See, if you remember triplet codon, I do not know how this has been told or not. If you scan this table, you will find that there is only initiated single codon, that is AUG, and there is single codon for methionine and tryptophan, 2, 3, 4, there is no 5. 6 codons, only important point is triplet codon, there is triple codon for 3 isoleucine. You note down this thing only, and 3 stop codons, and since there are more than one codon is present for a particular amino acid, this is called degeneration. That point you must remember. So the salient features of codon can come as a short note. You try to write down this thing, all 6 points, and the other additional points if you can remember. First and foremost, it is triplet, okay. And second is universal, it is same for all species, it is said. But there are exceptions, that exceptions I will tell you, and from that exception, you will might get an MCQ also. So mitochondrial DNA actually do not exactly code for the same thing. Second, this is universal and this is preserved throughout the evolution because these codons are applicable for both the prokaryotes and the eukaryotes. Only mitochondrial DNA differs. The mitochondrial DNA is having altogether a different translation machinery, which I will tell you in next class, in eukaryotic translation class. Degenerate means more than one codon, and non-ambiguous or unambiguous, one codon stands for only one amino acid, but there are more than one codons for particular amino acid. That is the degeneracy. And non-overlapping means it should be read in one direction from 5' to 3' direction, and non-punctuated means there is no comma. These other features are there. So you just note down here. This is the point which I want to highlight. Although it is said to be universal, and this is the exception. So UGA is basically a termination codon in cytoplasm for eukaryotes, but it is a codon for tryptophan in mitochondria. And this termination codon of mitochondria doesn't correspond with that of what we normally do, what a normal translational machinery uses in the cytoplasm. Maximum synthesis occurs in the cytoplasm itself. Only mitochondrial DNA uses those which are required for the respiratory exchange, that limited number of enzymes. That is the difference. So you remember this thing, UGA is a termination codon in cytoplasm, but it codes for tryptophan in mitochondrial DNA. So Uval phenomena, you know, although it is said to be universal triplet codon, but first 2 nucleotides are very much important. Third is relaxable. I will show you one example how relaxation is given to the third codon. The first two is very much important. That's why even if there are 6 codons, 3 codons, 2 codons, I think the first two are important. So this is the RNA codon table I am telling you. This is not a DNA codon table as I told you earlier. U is there, not T. So translation, basically we will tell today about prokaryotic translation. Unfortunately, I have checked last night your books, the common book which you read, that is Jambulkar's notebook. There are a few mistakes like initiation factor, there is no 4 initiation factor in prokaryotes. And in D.M. Vasudevan's book, I have seen it also. There is a mix-up between prokaryotes and eukaryotes, and particularly during the initiation phase, a few mistakes I have noted. Like initiation factor, what we say, that is initiation factor for prokaryote, we use it IF, initiation factor. For elongation factor, we use for prokaryotes, it is like this. Now, if it is for eukaryotes, then it is written for E. E. You will not see in the Dr. Vasudevan's book in the translation in prokaryotes, EIF 5 is written. That mistake you must take care of it. And in Jambulkar book also I have seen the initiation factor is basically for prokaryotes 3, 1, 2 and 3, not 4 and 5. It is not present. That you must take care of it, and proper functioning of that protein will not take place and there will be disease. So, protein synthesis must be understood very thoroughly, and to prevent bacterial growth, we have to use drugs, that is antibacterial or antimicrobial drugs. These drugs inhibit either replication or transcription or translation at different points. And these points you have to remember, there is no other way out. I will show you a few examples. Okay? And translation is a very energy intensive procedure. For each peptide bond, we require 4 high energy compounds that you will see. And also during initiation phase and elongation phase and termination phase, there will be 1 energy requirement. 3 more ATPs will be required. Basically, the GTPs are required for peptide bond formation. So that I will tell you. So there are 2 types of drug. One is called bacteriostatic drug. This I am not supposed to tell you at this stage, but I am just for your own clinical orientation I am telling you. Bacteriostatic drugs are those drugs which stops the growth. It doesn't kill. And these drugs, these bacteria or these microbes are left to our immune system so that we develop immunity. Bactericidal drugs are those drugs which kill the bacteria. These are given when it is very much violent, our body's immune system will not be able to take care of it, or if there is rapid recovery is required, then when bactericidal drugs are given. So there are different mechanisms. I am not going off antibiotic. It stops production of protein at multiple points through 3 main mechanisms. Okay. So this point you remember, RNA as an enzyme. Normally we all know, you just note down, write down this thing. This particular slide I want you to note down and try to understand. So normally all enzymes are protein in nature. The only exception is ribosome, this rRNA. This rRNA acts as a ribozyme. It is called, since it acts as a ribozyme. So basically snRNA I have told you during transcription. So snRNA forms spliceosome, and there is endonuclease activity and also splicing. There is splicing first and then endonuclease activity. Splicing means cutting. Okay. And then peptidyl transferase center is there, which I will discuss today. This peptidyl transferase center basically is endowed with the property of peptidyl transferase, which forms the peptide bond, and also it is endowed with the peptidyl tRNA hydrolase, which is basically an esterase which cleaves the peptidyl tRNA. That also I will tell you. So, you note down that in prokaryotes it is basically a 50S ribosome, which in the 50S ribosome catalytic part is only 23 rRNA, not the other part. Similarly, in the eukaryote it is only 28 rRNA, not the whole other RNA. That is the catalytic part. So, the catalytic activity is entirely mediated by ribosomal RNA, not by tRNA or mRNA. That you must remember. At least you try to write down these 2-3 points. These are the heart why RNA called is in a ribosome. So, translation of prokaryotes, I am trying to make it as simple as possible. So, you know ribosomes. Ribosomes are present, and I will first of all I will show you in a schematically, and then I will rapidly go through the slides so that you understand. So, ribosomes are present in the cytoplasm in the free state as well as bound state in the rough endoplasmic reticulum. Since there is no membrane bound structure in prokaryotes, so rough endoplasmic reticulum is not present. Ribosome is present in free form. So, ribosome is composed of basically 2 units. This is eukaryote, prokaryote ribosomes are basically 70S ribosome. Prokaryote, okay? Prokaryotic ribosome. This is 70S ribosome. Now, it is having 2 units. One is 50S, and this is larger unit, larger subunit. This is subunits. And a smaller subunit that is called 30S ribosomal subunit. Okay. Now, this 30S ribosomal subunit contains 16S rRNA and about 21 proteins. And this contains 23S rRNA, five S rRNA, and about 34 proteins. Actually, these proteins are mixed up with the rRNA. These proteins are globular proteins in nature, and some proteins are associated with a snake-like projection, so that this rRNA is positioned properly, kept in position, because that needs to be kept in position. So that is the ribosomal synthetic machinery. Ribosome is basically the synthetic machinery along with the mRNA. So, normally these ribosomes are in a free state. Remain this as subunits, okay, 30S and 50S. When they associate, I am telling big subunit, this is the small subunit. Okay, this is 50S, this is 30S. In the 50S, there will be 3 sites. One is A site, one is P site, one is E site. These are primarily located in the 50s larger site mainly, but when they combine together, they form the actual structure. Okay, normally they do not remain in this position; they are dissociated. So 30S ribosome contains 16S. Now forget about this thing. This 30S ribosome. This contains 16S, 16 rRNA. Okay, so this is suppose 16 rRNA. And this is an mRNA molecule in the cytosol. Now it is moving. This small unit, this 30S rRNA, scans the mRNA. Now, as I told you, in the mRNA, the transcription start site I have told you, and this is 5' UTR reagent. Now, you see the role of 5' UTR. The transcription will start from here, the synthesis, protein synthesis will start from this place. That is the starting codon. That is AUG. Basically, this 16S rRNA scans this mRNA. And just like Tata box, there is one consensus sequence, conserved sequence that is called Shine-Dalgarno sequence. You try to remember Shine-Dalgarno. Okay, this is basically a 6 base conserved bases. A, GG, A, GG. This is basically the conserved unit. In E. coli, U unit is also added. So that Shine-Dalgarno sequence is present in this mRNA. In the 23S, a 16S rRNA, this is SD sequence. Suppose this is Shine-Dalgarno sequence. So anti-SD sequence is there. Anti-SD sequence just complementary to that base. This base complementary to this base, there is one sequence here. So when it is scanning, it is coming across to this particular position. It is usually 3 to 10 nucleotide ahead of this transcription start site, that is the translation start site. Normally it is about 8 nucleotides ahead. So it indicates where the translation will start. So it basically scans, and then it basically, suppose this is the mRNA molecule. I am drawing this structure. Suppose this is A, GG, A, GG, U. Okay, suppose it is in E. coli. This is in mRNA. This is 5' end, this is 3' end. Okay, so this is anti-SD sequence is there in the 16S rRNA. So this will try to align, and this is the start codon. And this is the second codon. This is start codon, this is second codon. Okay, so it will scan, and that is a corresponding GCU, CCU. So this is present in, this is in the 3' direction, this is the 5' direction. This is rRNA. So rRNA scans and it places the anti-SD sequence against this so that there is a complementarity, and this forms an RNA dimer. Once this is formed, then basically this mRNA is held in position such that the starting codon is put in right place so that the starting codon can come and remain connected at the starting codon itself. So that is the importance of anti-Dalgarno, anti-SD sequence and the SD sequence. So Shine-Dalgarno sequence basically is just like Tata box of transcription. So once this is formed, the role of tRNA comes. tRNA basically carries the amino acid through its CCA arm. That structure I will show you. You know the tRNA structure. So this is the structure for CR. And this is an anticodon arm. So anticodon arm, this is the initiator codon AUG. It codes for methionine. Normally, the starting codon in eukaryotes is formyl methionine, not methionine. Formyl methionine. It is called F-met. So F-met, when it binds to the tRNA, it is called F-met tRNA. Normally, F-met tRNA will not be formed. tRNA needs to be activated. So for that activation, we require an ATP, and at least 2 high energy compounds will be required to form an activated amino acid. Once the activated amino acid is formed, then activated amino acid enzyme complex is formed, and that is catalyzed by amino acyl tRNA synthetase. Amino acyl, I will show you everything in detail. I am just telling you so that you remember amino acyl tRNA synthetase is the enzyme which basically forms the F-met tRNA. Now, once the F-met tRNA is formed, so that is positioned here. So, for initiation to occur, we require initiation factors. There are 3 initiation factors, IF1, IF2 and IF3. Okay, for initiation. There are 4 steps, main steps in translation. One is activation of mRNA, and then initiation, and then elongation and termination. So in every step, we require factors, and fortunately it is 3 in number. So initiation factor 1 is required for the initiation stage. For elongation factor, we require 3 again, EFTU, EFTS, EFG. 3 elongation factors will be required, and then at the end, when the termination occurs, or this polypeptide chain needs to be released. So that is called RF1, release factor 1, release factor 2 and release factor 3. I am telling you for you to understand the issue first. So what happens basically, normally this 30th subunit remains in this position where IF1 and IF3 remains bound. IF1 blocks the A site basically. It is the incoming amino acid site is there, so it blocks. IF3 prevents association with the 50th subunit so that both the subunits remain separate. So the basic function of IF3 is an anti-association factor. Anti-association factor. I will tell you all these things in detail. You just note down here. So basically, this is not. This remains like this. Then this carries the mRNA. mRNA is positioned. Now EF2, IF2 brings that F-met tRNA. And places it here. So IF2 basically, and once it is done, the IF1 basically makes this binding stronger. So the purpose is to position the initiator codon at such a place. So the initiator codon is the properly placed number 1. In this position, there is an incoming amino acid which will come. That site has to be kept free, and IF1 basically blocks this site so that no incoming amino acid can come immediately on the before the starting takes place. This is the initiation complex. So initiation complex basically forms the 30th ribosomal subunit, this mRNA, IF3, IF1 and IF2. Basically, there is a called ternary complex. You will read in the there is one term called. So first step will be activation of amino acid and then formation of the initiation complex. Activation of amino acid is done by the amino acid tRNA synthesis which requires ATP. And here ternary complex is formed, that is basically amino acid tRNA, that is activated tRNA, this IF2 and GTP. So there are 3 molecules, so it is called a ternary complex. This ternary complex then gets attached to it. So that is the F-met tRNA, so first amino acid attached to it. Okay, once this is done and GTP is there, GTP is hydrolyzed and then it associates with the 50th ribosome to make a complete ribosome. And now this is a cleft through which basically this polypeptide chain will be synthesized. So as I told you, this is the starting site, first amino acid. So this is called the P site. Remember, P site is the site where growing polypeptide chain will be attached to it, except the first one. First amino acid will be attached to P site and subsequent amino acids will come to the A site. This is for incoming site for all subsequent ones, and that growing polypeptide chain will attach to the P site, and after that synthesis has taken place, that peptide chain is formed, The RNA, there is one site called exit site. The tRNA which has brought the amino acid will be released and it will enter into the exit site from which the tRNA will be released. Okay. So starting taken place, now the initiation factor, all three will be required, all three will be with the hydrolysis of GTP, all factors will be released. Now initiation part is complete with the 30S ribosome, association with the 50S ribosome has taken place, and now the incoming amino acid will come, and that will be determined by what the nucleotide sequence is here. That will be determined. Now once this is done, elongation factor will come into the play. So all the three elongation factors will come. And again there will be a hydrolysis, there will be a GTP requirement, because this complex has to be formed, so for which one energy requirement will be there for initiation complex formation. Right? One amino acid will come here in the A site, okay, new site. Now I am showing it to you in a different way. So first amino acid has come, that is F-mate tRNA, F-mate, okay. Now in the inside of the polypeptide chain, there will be methionine, there will be tRNA, but that will not be formal methionine, that will be methionine, that you remember. Initiating mRNA, initiating amino acid will be F-mate mRNA, F-methionine, formal methionine. But other methionine inside the peptide chain will be methionine. Okay. Now second amino acid has come to the A site. This second amino acid has come to the A site as per sequence of the nucleotide. When this has been brought by tRNA, once this has come, this is the amino end, terminal end, and this is the carboxyl terminal end of the growing polypeptide chain. So amino group is here, so its carboxyl group is here, so its amino group is there, so it will form a peptide bond. Okay, and the tRNA will be first one, the tRNA is there, tRNA will move to the exit site. Now remember that, just open that ribosome site, that is a peptidyl transferase center. It is located mainly on the 50th ribosome. When 30th ribosome attached to it, the entire structure becomes complete. Now here the tRNA is attached to it with an ester bond. That tRNA will be cleaved by that peptidyl transferase center, peptidyl transferase enzyme. Okay, that hydrolase enzyme is there, ester hydrolase, that will hydrolyze, and the deacylated tRNA. Deacylated tRNA means free tRNA. Free tRNA will come into the E side, and from E side it will be basically coming out from the ribosome. So F-mate tRNA there will be peptide bond. So suppose this is the second amino acid. Second amino acid will be attached to it, and then these two amino acids, this amino acid, this will be linked to basically tRNA. This will be moving from this side to this side, because this is the chain of RNA. This is the codon, next codon, suppose XY. This RNA will translocate one codon ahead. Once this part is complete, a dipeptide has formed, a dipeptidyl tRNA has formed. That means the purpose of that second codon is over. Now this ribosome will move further, so that the entire this exit side is now open, From that exit side will go out the tRNA and the entire structure will come one codon forward. That is called translocation. Okay, and the same process will occur. Again, the peptidyl transferase is there in the peptidyl transferase center, that will form a peptide bond. Again, the tRNA will be removed by the esterase hydrolase. It will come to the exit site and it will be released. And this will continue like this until we reach a stop codon. Is this process clear? This elongation phase, there will be requirement of the 3 elongation factors. The functions I will tell you in a slide that you will note down. What this RF, this termination codon has come, that is termination codon. There is no tRNA for the termination codon. So nothing will come, this will stop here. Now here the release factor will come, that is RF1, RF2. RF1 will basically, there are 3, UAA is universal, it can bind to UAA, basically remember UAG and UGA or UAA. RF1 binds to termination. RF1 binds to UAG, RF2 binds to UGA. So RF2 will bind at the end of the termination codon to this site. Now that RF will not code for any amino acid. So that is will attach to it, and that will help, this RF release factor will help the peptidyl transferase center to cause hydrolysis of the amino acyl tRNA, to release tRNA, so that the polypeptide chain will be released and the tRNA will be again deacylated, means it will be free. This will go into the P site, and then the RF2 or RF1 will remain in that site. Now to remove that RF1 or RF2, RF3 will be required. Now, RF3 will, in association with ribosomal factor or disruptive factor, will dissociate the bound, that is 50S and 30S ribosome, complete ribosome to dissociate. So everything will be released. The growing polypeptide chain, the release factors, tRNA, and the ribosome which has been united, all these things will be free to make a second cycle. Now I will tell you briefly in detail how this is done. Now when basically normally it is in the free state, now when there is a deficiency in the environment, so bacteria will not be able to synthesize protein, then it will remain in that dimeric form, that is 50S and 30S form. These 50S and 30S form will be present when there is a active translation is going on, and when it goes into hibernation, means when it is depleted with the nutrient, and there is 3 factors which will also keep that in that position. These are ribosomal modulation factor, hibernation promotion factor, and also ribosome silencing factor. These are the 3 factors which will keep the ribosome in that position at original ribosomal that is 50S and 30S, that is 70S ribosome formation only in starvation condition when the nutrient, the environment is starved with the nutrition. So, a ribosome cannot occur. So, only thing is that the energy requirement will be there at 4 steps, 2 energy will be required for each peptide bond. 2 high energy phosphate bonds will be required for the activation of the amino acid. One will be again required for the initiation complex, one will be for the elongation, and one will be for the release factor. So, this will be the basic energy requirement. Remember, for peptide bond formation, remember one thing, for peptide bond formation, no energy is required because amino acid is already activated. So, peptidyl transferase enzyme, which is in RNA, basically rRNA, will catalyze the formation of peptide bonds without requirement of any energy. Okay. So, since this will be amino acid will combine with ribosome, this amino acid hydroxyl group, so one water molecule will go out. Similarly, when the esterase will break, it will require one molecule of water. That one molecule of required water will be provided by the release factor to the peptidyl transferase center. That you remember, that hydrolysis will take place by the provision of one molecule of water by the release factor to the peptidyl transferase center to peptidyl amino cell tRNA hydrolase enzyme. So, this is the gene. Now, I will show you very briefly in detail. So, that what is translation by this time you know, this is basically a conversion of nucleotide sequence into an amino acid sequence, nucleotide sequence of the mRNA into amino acid sequence of the protein. So, it will be polypeptide in nature. Normally, one tRNA is attached to it and it scans. Actually, in bacteria, it is polycystronic, means there is multiple, but remember that there is a size of ribosome. Ribosome is not a simple structure. So, at least 35 nucleotides apart, another ribosome can attach to it, not less than 35 nucleotides. This can happen in eukaryote also, when there is a rapid translation will be required. So, polyribosome means ribosomes attached to a single mRNA molecule and translation is taking place from the same molecule, mRNA molecule, from multiple places at a time. Okay. So, the first amino acid will correspond to the, that is the amino terminal end, and the last one will be the carboxy terminal end. So, mRNA doesn't have any affinity for amino acid and it will not bind to amino acid. Only thing is that if that Shine-Dalgarno consensus sequence of the mRNA combines with the anti-S sequence of the rRNA and the initiation factor 2 brings that activated tRNA amino acid, then only it will, the amino acid will start, translation will start. So, that requires an adapter molecule and that adapter molecule is basically provided by the tRNA molecule. So, this function of the tRNA molecule is to bring that amino acid. Functional groups are of the amino acids are not in contact with the mRNA molecule at all. The functional groups are a little bit ahead, however, only it is positioning the amino acid. Now, the amino group and carboxyl group are present at a different site. So, this is the initiation phase, as I told you, IF1 and IF3, it will be attached to the 30th ribosome, 50th ribosome is separate. Now, this is the activated amino acidyl tRNA, it will come, this is the 16S rRNA, and this is the mRNA, the antiS sequence is there. It is forming an RNA-RNA dimer, and then initiation stage continues. The F beta, this is the initiation factor 2. This combined share, and that is forming a 30S. This is the attached till it is attached to the 30S unit, and once this attached to it, then the 50S subunit will attach to it to make a complete thing, and there will be narrow tunnel through which the mRNA will pass. As this large subunit, I am telling you, this is not endowed with the property of proofreading, but there will be conformational changes in the 50th subunit, and if by chance a false and wrong incoming amino acid comes, it will cause a conformational change. This 50th ribosome will make a conformational change, and it will reject it. And then when the right amino acid will come, then only it will allow. So that is called conformational proofreading. Remember one thing, conformational proofreading occurs in prokaryotes, and that is done by the 50th ribosome subunit. And all the three sites, that is P, A, P and E sites are present primarily on the larger subunit, but when the two units combine, they form the complete structure. So now you see, this is the A site. So this was attached to the P site. P site is attached, I am telling you again, that will be for the initiator codon. But all other subsequent amino acid will be positioned at the A position only. That is called incoming amino acid, tRNA. And but when the peptide bond will be formed, again that peptidyl tRNA will be attached to this P site. That's why this is called P site, peptidyl site. That is A site called amino acyl tRNA site. That is incoming amino acid tRNA site. This E site is called exit site. So elongation will take place. So this is the elongation take place. These two amino acids, as I told you, this peptide bond will be formed here. So peptide bond is there. Now P tRNA is there. tRNA will come out from the exit site, and there will be growing polypeptide chain which will be attached to the P site only. So this P site chain will come, this will end, and at one point of time it will come in contact with the termination codon. So the release factor will come, that RF1, RF2 I have told you, or RF3. This RF1 and RF2 will be attached to it, and basically then RF3 will detach. Now the entire structure is detached with the ribosomal disassembly factor. So you see that this is the polypeptide chain, the two subunits are detached to it. So this is the structure of the tRNA. It can come as a short note. It is having 4 arms, and these 4 arms are having different function. This is the anticodon arm, which combines with the mRNA as per sequence. This carries a specific mRNA. Now only the difference between the F-mate tRNA and the tRNA is that at the CCA arm there is an unpairing, nucleotide unpairing, and this anticodon arm is endowed with G-series region. These are the two differentiating points between the F-mate tRNA and the tRNA. You understand? F-mate tRNA, there is no nucleotide pairing at the CCA arm, that is in this arm, and the G-series region is present in the anticodon arm. That differentiates. Okay, so the functions of the anticodon arms, you must remember that anticodon arm will come in contact with the this mRNA, and this site CCR will carry the amino acid, and this is called the UDHU loop. That is called, this will recognize the amino acid tRNA synthesized, and this will recognize, this will fix that site to the tRNA. This is ribosome. So as I told you, now show the oobleing. See, CUC is the mRNA sequence here, but GAG is a common identical lives in tRNA. So you see the GAG, this is lives in tRNA. Now this is a normal pairing. Will take place here. Hard codon is not important. So this is oobl pairing. This is G is normally will combine with C. Now you see the G is there, but it is combining with the U. that is called an ovule pairing. That is one fallacy of that 61 RNA codon. So as I told you, this RNA, these two units of RNA, when they will combine, they will form a cleft, and they are mostly, the RNA proteins are mostly globular in nature. Some are having snake-like projections that we have seen. So the catalytic portion is present in the deep cleft, that peptidyl transferase center, in the deep cleft, and through which the mRNA runs. Okay, so now by this time you know that this all I have explained, that 50S ribosome is having 23S ribosome, 5S ribosome, and about 34 proteins. 30S ribosome is 23S ribosome, that is having an anti-SDA sequence, and that maintains complementarity. So the catalytic site is basically 23S rRNA. Again I am telling you, this is the translation apparatus. So basically I am not showing it in the E site here. So 50S and 30S rRNA is formed. Now this is the growing polypeptide chain. P and S site assembly will be formed when it will be complete. Ribosome basically ribosome, mRNA, tRNA, and the elongation factor, initiation factor, and elongation factor. Okay, and the release factor also. So three sites are there. As I told you, this is the outgoing tRNA. So A site is the site for entry of the incoming amino acid. P site is for the growing polypeptide chain, and E site is for the outgoing tRNA. So translation will continue until it will. This is the structure of the polypeptide chain based on the mRNA sequence. The length of the polypeptide chain will depend basically, and this is the termination codon, that is the mRNA requirement. So once this everything is complete, that polysome, as I told you, there are many ribosomes will be attached to a single unit of mRNA, that will form a polyribosome. And when the synthesis is complete, then everything has to be released into the cytoplasm, so that another cycle of translation can take place. Okay. So requirements, as by this time you know that, as I told you, that three release factors, the three initiation factors, IF1, IF2, and IF3, three elongation factors, three release factors. Okay. And regulatory factors, as I told you, are ribosomal modulation factor, hibernation promotion factor, and ribosomal silencing factor. These are the three factors which will down-regulate the translation synthesis, because they will keep either the 50S subunit as such, or they will not allow it to bind between 50S. This is ribosomal silencing factor that basically prevents from joining the two units. So step 1, as I told you, that amino acid will be activated and it will require ATP. Remember, here AMP is formed. That means two high energy compounds are required. So that is the activated amino acid tRNA. So this will be basically first and foremost. So first an enzyme amino acid AMP complex will be formed, and then basically that tRNA, activated amino acid tRNA will be formed. Now this is the basic actual reaction. So it will basically take place in two steps. This will get in the book. Now dissociation, as I told you, IF1, IF3 with the 3S, 30S unit, third unit I have explained, MBA tRNA, IF2 and GTP, then formation of 30S initiation complex. Once this is formed, then 50S will combine and to form a 70S complex. And this you note down. This is not present in your book. You please write. The three things you must write. These are the, otherwise I will not. The four points you must note down. These are not present in your book. So functions of the initiation factor. Try to remember what I have written in the, at least in blue, so that you can remember. Rest, if you cannot remember, write down at least one point. These functions of the initiation factor, functions of the elongation factor, function of the release factor, and what the GTP is required. These are the 4 slides. You have to write down. Just take 5 minutes extra time. Rest of the things you will get in the book. Or should I go through? You can note down. Later also I can, in a SGD class also I am saving the time because you will have to go. So if the two subunits are joined together, there will be no basically synthesis. This needs to be separate. So inhibitors of potency prokaryotes, you must remember in which step it basically blocks. So Shine-Dalgarno sequence I have explained, and that is present in the 16S rRNA that I have also explained. That positions that I have explained in detail. The recognition is basically done. This is the stop codon, this is the start codon, it is little ahead in the 5' transfer region. When there is a leaderless, that means when there is no 5' UTR, it will cause a leaderless initiation. Means that 70S ribosome will start initiation. That you will note down, that leaderless initiation is also possible in prokaryotes. So pre-initiation complex, as I have told you, that when these things, everything will be combined, then GTP will be hydrolyzed and all three factors will be released. Now this is the P site, A site, the inhibitors of ribosome synthesis, you must note down. This I have already explained. Now functions of elongation factor. There are 3 elongation factors. You have to note down the elongation factor again, or otherwise I will give you time later. So elongation step will continue. Translocation will occur by one codon at a time, and this will be very fast. In transcription, at least 1000 nucleotides can be added per second, because there are 4 nucleotides only, but here 20 new amino acids are there. That's why the synthetic machinery of protein translation is very slow, about 25 to 30 per second, not more than that. In contrast to 1000 in eukaryotic system, it is still slower. So elongation step is there. So once the elongation is complete, there will be again inhibition at this stage. Termination, as I told you, RF1, RF2 and RF3. It recognizes, remember, RF1, A is first capital letter. So try to remember A is the release factor 1, it will recognize, and UGA, G comes after that, so that RF2 will recognize UGA, UA is common, and to release either RF1 or RF2, RF3 will be required. Okay. So this will be the ribosomal disassembly post-termination complex. So ribosomal recycling factor will be there. That will basically disassemble. Ribosomal disassembly factor I have this will be ribosomal recycling factor. Ribosomal recycling factor. So ribosomal recycling factor will be there. So that will basically dissociate into all ribosomes and a fresh ribosome cycle will occur. So inhibitors of protein, this will act here. So GTP requirement, at 3 places. This also you try to remember. Down regulation, I have told you there are 3 factors which are required when it is deprived of nutrients. That ribosome modulation factor, hibernation promotion factor, and ribosomal silencing factor. These are the 3 factors which keep the translation at fault, because there is not adequate amino acids which are available for protein synthesis. So what I want that you make a tabulation, inhibitors of replication, inhibitors of transcription and inhibitors of translation. Make a chart before the entering into the examination hall, 1 hour before the examination hall. You try to have a look at it. Otherwise, you will not be able to remember by reading once. You make this chart at your convenience, at least you will be able to write down something if a short note comes or if there is an MCQ. Which step is inhibited by that procedure, that you will note down. You can make in your own, this I have made. You read from the books, note down all the 3 parts, make a table. The way you can remember easily, because you have to remember many things, and this you keep reserved, something just 1 hour ahead of the examination, so that you remember. Otherwise, you cannot remember, honestly speaking. I also failed to remember. So there is a video, because of shortage of time, I cannot show it to you. Ample number of videos are available. First, if you cannot understand, those who have understood, that it will be easier if you see first a video. it will be 3 to 4 minutes video or even a 5 minutes video, it will be sufficient to understand it first, how it is happening. Then only thing is the rest of the theoretical part you have to memorize. You first remember what are the steps, how it is done, and then you go through all the theoretical part behind it, particularly the functions of the 3 factors, initiation factor, elongation factor and release factor, and GTP requirement, that is energy requirement of translation in prokaryotes. Eukaryotic synthesis translation will be very large class, so have patients read it thoroughly so that I can go through very quickly. These steps are similar, only thing is that the transcription factors, elongation factors are different, nothing more than that. Basic steps are similar. So once you read it, then only you will be able to understand. Okay. I expect that you must go through it, and while reading the book, you be very particular. There is a mixture. 3 books I have seen, Harper, I am telling you, it is not written properly, mixed up. And prokaryotic translation has not been written at all in the 32 edition. I have checked last night. So eukaryotic transcription translation is required very nicely. In Dr. D.M. Vasudevan's book, there is a quite mix up between the two. It is very difficult to understand. Without this class, it is practically impossible, and all these things are not written at all.